Hi, my name is Arisa Coombs, and today I'm going to be talking about CRISPR. CRISPR is currently the most advanced as well as the most user-friendly technology in the realm of genome editing today. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is a mouthful. Clustered Regularly Interspaced Short Palindromic Repeats essentially just means little chunks of DNA that are inserted into one long strand of DNA where they don't belong, which can deactivate a gene. This technology was originally discovered in E. coli bacteria in the 1980s. These bacteria use this as a defense system against viruses. When the bacteria encounters a virus, it can incorporate part of the virus's DNA into its own, which it can use later to recognize the virus when it comes back. It can then insert those chunks of DNA that I talked about earlier into the virus's genome, which it can use to deactivate the virus, essentially killing it and protecting itself. However, this is useful not just for protecting bacteria from viruses, but it can also be used to target very specific genes in other types of cell, from bacteria to human. One example of this is at Stanford University, where scientists took stem cells in mice in order to correct sickle cell anemia. They reprogrammed stem cells to produce healthy blood cells instead of the sickle cells, and when injected back into the mice, these healthy cells rapidly outstripped the number of sickle cells, which solved the problem. Scientists are also moving on to use CRISPR in humans. Recently in China, it was used for the first time in a cancer patient. Essentially, white blood cells were reprogrammed to act the same way as the E. coli bacteria does when it uses CRISPR. These new white blood cells can recognize the DNA of cancer cells and attack them when they are encountered in the bloodstream. However, CRISPR also has ethical issues because of its potential applications. Once again, recently in China, CRISPR was used in artificial fetus cells. However, it was found that CRISPR caused more mistakes than it did in other types of cells when used in the fetus cells. CRISPR actually inserted DNA chunks in places where the scientists had not intended them to be. This problem could create new types of diseases that we have never seen before that could be passed on from generation to generation. However, because of CRISPR's potential to solve diseases like sickle cell and cancer, it should continue to be pursued and developed because of its ability to improve the quality of life for people all around the world.